biology. Today we're talking about respiration. So respiration happens in the mitochondria, and the mitochondria has this overall structure where it has an outer membrane and it has a very highly folded inner membrane. And as I've said many times, anytime there's a folded something in biology, it's to increase surface area to volume ratio. So the matrix is the part of the mitochondria that's basically like the cytoplasm. The cristae are these folds, and then it has an inner membrane and an outer membrane. And then this space in here is called the intermembrane space. Okay, so in that inner mitochondrial membrane, pyruvate gets turned into acetyl-CoA. And the reaction happens like this. There's a coenzyme called coenzyme A, and it is going to attach to an acetyl group through this sulfur here. So what happens is, as the pyruvate gets moved across the membrane, we're going to lose a CO2. The carbon dioxide is going to bubble off and get eliminated from the body. We're going to harvest an NADH, and we're going to attach that coenzyme A to that acetyl group. So you wind up with acetyl-CoA. Let's see. Remember that for every glucose, we're going to do this twice. So we're just showing it once here, but for a glucose, you're going to do it twice. Okay, that acetyl-CoA then winds up taking that acetyl group and attaches that acetyl group, so 2-carbon group here, attaches it to a thing called oxaloacetate. It does not really matter that you know the names of these, because you're always going to see, you're always going to be presented with a diagram. So you need to know how to interpret the diagram. So what we see here is an acetyl-CoA attaching to this oxaloacetate here, and that acetyl group gets attached, and now you start with, now you wind up with a 6-carbon compound. So that 2-carbon compound, acetyl group, gets attached to the 4-carbon compound. You wind up with a 6-carbon compound, and then the coenzyme A goes away and can pick up another acetyl group. Okay. In the next step, we're going to see that citrate get turned into isocitrate. Not a step you really need to look much detail in. But then here we see isocitrate going to alpha-ketoglutarate, and if you remember, anytime we have an NAD plus going to NADH, we've harvested a pair of electrons. So at the same time we're harvesting electrons, we're also cleaving off a carbon dioxide, which drives this reaction forward. So we harvest electrons and we get rid of a CO2. And then in the next step, look what happens again. We're going to harvest another pair of electrons, and we're going to get rid of a carbon dioxide. So now we've lost as many carbons as we put into this cycle. So then now we have 4-carbon compound that we're going to regenerate or change back into oxaloacetate. In this step down here, we make a GTP, which is energetically the same as making an ATP. By substrate level phosphorylation. And then we harvest another pair of electrons. So now FAD is like NADH or NAD, but because it is an electron carrier. So when we go FAD to FADH2, you're just harvesting a pair of electrons that you're going to do something with later. Okay? And then we do some more rearrangements here, and we wind up harvesting another NADH, so another pair of electrons. So if you look at this whole thing, we harvest 1, 2, 3, 4 pairs of electrons. So 3 go onto NADH, 1 pair goes on to FADH2, and we also make a GTP. And remember, because we started with a glucose back in glycolysis, we chopped that into 2 3-carbon compounds. We're going to run this cycle twice for every glucose. Okay? Now, so at this point, it looks like we've only made a GTP. We haven't really gotten any energy storage in a form that we can use. But these electrons on NADH and FADH2 can be used. And what they're used to do is to move hydrogen ions, or sometimes people will call them protons. You'll hear books call them protons, or basically a hydrogen ion is a proton because it's a proton with no electron. That's hydrogen. And what we're going to do is we're going to feed them into the electron transport chain. Okay, and so if we feed those electrons on NADH into this complex 1, complex 1 can use those electrons to attract the proton and pump that hydrogen across the membrane. Basically what it does is the hydrogen's attracted to it, and then it dumps the electrons off to another molecule here called quinone, and that forces the hydrogen ions to go across the membrane. The electrons that are now on quinone can be passed off to complex 3, which can pump another hydrogen from the mitochondrial matrix into the intermembrane space. So same pair of electrons now. They're going to get passed off to cytochrome C, which is going to pass them off to complex 4, which is going to pump another hydrogen ion. Complex 4 now has a pair of electrons that it needs to put somewhere. And where's it going to put it? Well, if you take those electrons and you attach them to an oxygen, you can make water out of them. And so you attach those electrons to oxygen, you make water, now the electrons are gone, and this electron transport chain can keep moving electrons down the chain, and by moving electrons, it can pump hydrogen ions or protons into the intermembrane space. So you wind up with a lot of hydrogen ions in here in the intermembrane space, very few out in here in the mitochondrial matrix, and notice that when you make water, you actually get rid of some hydrogen ions here because you're attaching them to water. Okay, so there's a pH gradient here. The other piece, remember, we harvested electrons on FADH2. FADH2 holds those electrons a little more tightly than NADH, and it can't dump those electrons off into complex 1. So it actually dumps its electrons into complex 2. The electrons go into quinone. Quinone passes them off to complex 3, and complex 3 pumps a hydrogen. They get passed off to cytochrome C, passes them off to complex 4. Complex 4 pumps a hydrogen. So when you use an NADH, you can pump all 3 of these hydrogens using complexes 1, 3, and 4. When you just use FADH2, you bypass complex 1, so you can't pump as many hydrogens, but you can still, pump some. All right, so that is basically what happens in the electron transport chain. So now we've set up this concentration gradient, and if you remember chemiosmotic phosphorylation, we can use the ATP synthase to harvest the energy that we've stored in this intermembrane space by putting an ATP synthase across the membrane, allow those hydrogens to go down their concentration gradient, spin this turbine here, and force a phosphate to go on to ADP, making ATP. And that's basically what respiration is.